it is the preponderance of the evidence clearly now points to a lab leak. The evidence is of 4 different kinds. There is molecular evidence, that is to say the sequence of the genome of the virus suggests editing of a kind that we know was taking place at the Wuhan Institute of Virology. There is phylogenetic evidence, that is to say the group, the sarbecoviruses to which SARS-CoV-2 belongs are known not to have furin cleavage sites, and yet this furin cleavage site shows up phylogenetically out of place in SARS-CoV-2. There is epidemiological evidence. We have a virus that did not show any evidence of evolution early on. It arrived in human beings perfectly adapted to spread at a high rate, whereas we would have expected a period of evolutionary adaptation that we cannot find. And then fourth, and maybe most important, there's a tremendous amount of circumstantial evidence surrounding the way the Wuhan Institute of Virology has dealt with the question of a possible lab leak. Initially, the laboratory in question acknowledged that a lab leak was possible. In fact, the leader of that lab, Zhizheng Li, said she didn't sleep a wink for some time, thinking that maybe the virus had escaped from her lab. So we know that such a thing is plausible. We also have the case of them having renamed a sample in a way that contravenes scientific protocol. So RATG13, which is the closest relative of SARS-CoV-2, turns out to have had a prior name in that laboratory, 4991. There's no reason to have renamed it. It effectively suggests keeping of 2 sets of books. So in any case, all 4 types of evidence point towards a laboratory leak that is now being obscured. When we say that the virus may have been modified in the lab, we are not necessarily talking about edits where someone sits down to introduce a sequence. That is certainly possible. That is something that we do. But we are in general talking about a type of gain-of-function research that uses a process called serial passaging, in which we do things in the lab that we literally do not know how to do. And the way we do it is we use evolution. We take a virus and we can introduce it into tissues or into animals, and then we can allow it to transmit between cells or between individuals of these animal species. And so evolution does the work for us. And for anyone to argue that because, as the initial Nature paper that dismissed this possibility argued, although SARS-CoV-2 is very good at transmitting between people, the sequence is one humans would not have predicted would be successful. That was the argument against the possibility of this being human modified. But it elides the strong possibility that serial passaging discovered things about transmissibility that we humans simply did not know until it had occurred.